Dear Mr. Dorin Grecean, Prime Minister of the Republic of Moldova, dear Professor Dorin Grecean, Director of the Technical University of Moldova, His Excellency Mr. Yojiro Yamata, Ambassador of Japan to the Republic of Moldova, dear Professor Randall Sherman, University of California, Berkeley, Nobel Prize laureate, dear Professor Nicolae Palikaratis, International Federation for Medical and Biological Engineering, dear Professor Emil Jaban, Rector of the State University of Medicine and Pharmacy, Nicolae Testemitanu, dear Professor Dr. Shomtia, co-chairman of the International Conference on Nanotechnologies and Biomedical Engineering. Dear attendees, ladies and gentlemen, good morning. It's a nice day in Moldova, 20th of September, good weather, and on behalf of the International Advisory Committee, on behalf of the program and organization committees, I would like to welcome you to the 6th edition of the International Conference on Nanotechnologies and Biomedical Engineering. So we started to organize this series of conferences in 2011, 12 years ago. With the time, we gained experience, and more people, more and more well-known researchers, professors from many countries of the world started to attend this conference. And I would like to say that indeed it became a very strong international meeting where researchers from many countries meet, discuss, exchange opinions, start to prepare and to promote joint projects. And Moldova became stronger also through the organization of this conference. Of course, thank you very much for your acceptance to attend the conference in spite of the fact that the coronavirus is still in the air and there is a war in our region, in the neighboring country. In spite of all these barriers, you are here. And this reflects your high interest to the scientific research in general and interest in your desire to meet our researchers and to discuss joint results and to start new joint projects. Ladies and gentlemen, it's my pleasure to give the floor to Mr. Dorin Grecean, Prime Minister of the Republic of Moldova, who will come with a welcome message. Excellency, you have the floor.
Thank you very much. Look, I'm extremely honored to be among the best intellectuals, not only from the Republic of Moldova, but from elsewhere on this planet. I'm only very happy to welcome our scientists, but also the scientists from abroad. And I know that Moldova can contribute, and now with globalization, it's even more important to have this collaboration. So I'm only very happy to see this conference having taken place every year in Chisinau. I'm also very glad that we are aligned with the future. The way I see the future, which is becoming extreme actually, is that we have to advance in 4 fields. The first is the material science. And here we are looking into more and more resilient, flexible, and other types of materials materials that allow us to prosper. The other thing in the material science is how we can build things and materials not only on our planet but elsewhere as well. And here we have progress. The second field is the life sciences. And here the people want to live longer, want to live healthier, and the science has an answer to that. The third field is the computer science. And here people want to have the replication of our brains, how we think, and this is our brains is very efficient, and correspondingly we are looking into how to replicate this. Machine learning is machine learning, artificial intelligence is still something to be achieved. And the fourth field is the human talent. The human talent at its best use and best development. I'm very happy to see that the conference addresses directly the first two fields and contributes with the other two fields indirectly. I wish you success. I saw the program. It's very intensive. I wish you all the success, and I'm sure that at a certain point in time, some industrial or human life applications will come out of the work of today's conference. Thank you very much.
Thank you. Ladies and gentlemen, it's my pleasure to give the floor to Professor Vera Bostan, Director of the Technical University of Moldova. Please.
Distinguished participants, ladies and gentlemen, a very good morning to you and welcome to Chisinau. And we are very honored to be co-organizers of this important conference, a conference that already is marked by high achievements, bringing together scientists not only from Moldova but throughout the world. I'm very also honored to have here with us a remarkable scientist, Professor Andy Sheckman from University of California at Berkeley, whose roots are from Bessarabia. I'm very happy to have all friends again, despite all the problems we have in the regions, who came to Chisinau. I would like to thank the organizing committee and the scientific committee for their dedication and work and competence in setting up this forum, and also to thank in a special way Professor Victor Shontea, who is one of the founder fathers of this conference and whose efforts actually brought us here in Chisinau today. Dear colleagues, I'm sure that this conference will contribute to the development of the research not only in Moldova but throughout the world. And this conference is a special one because, as Mr. Prime Minister mentioned, here we have two important fields which today represent one of the driving motors of change. Those fields are nanotechnologies and biomedical engineering. And in the same manner as semiconductor technology reshaped the previous 70, 80 years, today I'm very confident that nanotechnologies, material science, and biomedical engineering, biomedicine in general, will reshape the years to come. I wish you success in the works of this conference, and I'm hoping that this conference will contribute to the development of science in the Republic of Moldova, and not only will enhance our cooperation and will lead to new projects. I wish you good luck, and thank you very much for your attendance of this important forum.
Thank you. Ladies and gentlemen, I would like to give the floor to His Excellency Mr. Nicolae Yamada, Ambassador of Japan to the Republic of Moldova. We have many guests, many professors from Japan. It's a tradition already that we have excellent teams attendees from Japan. So, please, you have the floor.
Good morning, everybody. Thank you, dear Prime Minister. Thank you, dear President, the Academy of Sciences. Thank you, and good morning. And my name is Yoichiro Yamada. I'm ambassador of Japan to the Republic of Moldova. I came here about 9 months ago and trying to understand what this country's strengths are and how, as ambassador, I can be a bridge between the Republic of Moldova and Japan. And then one of the things that I found was that the Moldova is very strong in nanotechnology. I was really surprised and impressed when I paid a visit to the Technical University of Moldova, and then he was shown around to be acquainted with the achievements that university has made. And I'm really honored to note that there are distinguished professors from Japan. And then this is certainly an area most advanced in making our humanity closer to a greater or higher levels of prosperity. Talking about nano, there was a nano satellite launched into orbit, which was a small satellite manufactured by the Technical University of Moldova last year. And then the launch pad was a Japanese spacecraft. So here was another example where nano brought Moldova and Japan together. But science is an area where international cooperation, not just bilateral cooperation, but a really multilateral international cooperation makes sense and has made sense for all these years. And it's really exciting to see so many distinguished researchers, professors from all over the world to be present here, and I'm very honored to be invited to this podium today. Thank you.
Thank you, Your Excellency. Ladies and gentlemen, I'm honored to give the floor to Professor Eddie Sheldman from the University of California, Berkeley, Nobel Prize laureate in medicine, a good friend of the Republic of Moldova, honorary member of the Academy of Sciences of Moldova, and Dr. Honoris Causa of the State University of Medicine and Pharmacy, Testemitanu. Professor Sheldman is in the Republic of Moldova starting from Sunday evening, so already has some impressions, and actually, as far as I know, positive impressions, and I am happy to give him the floor.
Thank you, Professor Ticignan. Ladies and gentlemen, I'm delighted to be here. This is now my third day visiting Moldova, though the trip has been long planned. Professor Ticignan first approached me several years ago after he learned that in the biography that I posted online, I said that my grandparents came from Bessarabia. And so naturally there was an affinity, and I'm delighted that he reached out to me. My family back in the States is quite excited about learning about this trip, and I'll be the first person to return to this area in nearly 100 years, because my grandparents emigrated in 1927 from the then village of Lipkan, which I will return to see tomorrow, so I'm very excited about that. I've had wonderful opportunities in the first 2 days to visit the Technical University and at the Medical University, and to visit with the faculty and share my thoughts. I'm impressed, just in the brief time that I've been here, at the urgent desire to advance as a scientific society emerging from years of control by the Soviet Union and still the shadow of Russia now being put aside. Moldova has a tremendous opportunity to exploit the natural talent of its young people, but it must invest even more in science so that the young people who are trained in universities here have an opportunity and are encouraged to remain to develop not only the intellectual basis of this country, but also the industrial base. I'm eager to help. We visited with a parliamentarian yesterday, and I see a path forward, and I'm eager to share my own research with you this morning. So I'm delighted to be here and look forward to meeting more of you.
Thank you. Ladies and gentlemen, it's my pleasure to give the floor to Professor Nicolae Palikarakis of the International Federation for Medical and Biological Engineering. Professor Palikarakis is also a friend of the Republic of Moldova. Please. Doctor honoris causa of the Technical University. Please, you have the floor.
Thank you very much. It is my honor to be in this Congress again. I think that it is in the majority of the organization of this conference I have been present, and this is due to the fact that 15 years ago I met Victor Sondea, and through two big projects of Tempus in biomedical engineering education, we created very strong links. But it's also an honor because it's the first time actually that I'm in a conference that the Prime Minister of the country is present, and I'm so glad and grateful that you recognize the importance of biomedical engineering by your presence here. So today and this time I'm going to address this Congress on behalf of Ratko Majarevic, who is the President of IFMBE, and I have to give you some information about the International Federation of Medical and Biological Engineering. Okay, we have the... Okay. So IFMBE, who is the International Federation of Medical and Biological Engineering, has been created more or less 60 years ago with main mission to encourage, support, represent and unify biomedical engineers all over the world in order to support better health applications for the benefit of patients. IFMBE is the largest biomedical engineering organization in the world with many national and international partners. It has been created in 1959 in Paris and has been firstly registered in Paris in 1959, I think. Today we have a very complicated, I would say, but very large organization with its administrative council that Ratko Majarevic is the president right now. There are ordinary members of this council, there are regional groups, representatives in the council participate as well, and also there are 3 divisions of IFMBE representing very critical sub-specialities of biomedical engineering: clinical engineering, health technology, assessment, and digital health very late. I had the honor and the chance to be chairman of Clinical Engineering Division and the Health Technology Assessment Division, and also some supporting bodies, committees, etc., that support the general function of IFMBE. Here you see a map on the different regions and countries that are represented in IFMBE. IFMBE is part of the IUPESP together with the International Organization of Medical Physics, and through IUPESP it is very well connected to United Nations Organization, to International Science Committee, and also to standardization bodies. As concluded, in order not to be so long, since the start in 60 years, 65 almost years ago, has grown very significantly, and today it represents the most large organization in biomedical engineering. Unifies biomedical engineering community, and as IFMBE, we appreciate the effort by all our members to participate and support these actions. So it is essential that all the societies are supporting the role of and the tasks of IFMBE, and we are very glad and grateful to Professor Soltea and the Moldovan Society of Biomedical Engineering for the support that they give. So thank you very much again. It is my pleasure to be here and address the way that this conference will conclude.
Yeah, thank you. Thank you so much. So I would like to give the floor to Professor Nicolae, Rector of the State University of Medicine and Pharmacy.
Good morning, everybody. Dear Mr. Panelist, dear guests and colleagues. Biotehnologiile, tehnologiile medicale, ingineria, astăzi este o necesitate. Noi acest lucru l-am analizat și sunt foarte mândru și bucuros că participăm ca coparticipanți la această expoziție. Astăzi, medicina nu poate fi fără tehnologii, fără tehnologii noi. De aceea, eforturile comune și împreună cu domnul rector Bostan avem planuri pentru a dezvolta aceste programe noi, a introduce în sistemul educațional pentru a implementa în sistemul de sănătate în Republica Moldova. Vă mulțumesc toți pentru participare și sunt mândru că avem un număr foarte mare de lucrări la acest eveniment. Mulțumesc!
Professor Nicolae told about the importance of nanotechnology, biomedical engineering, biomedicine in general, and of course he is very happy that attendees from many countries are in the conference hall and wishes the best to all attendees. So, ladies and gentlemen, I would like to give the floor to Professor Victor Șontea, co-chairman of the International Conference on Nanotechnology and Biomedical Engineering. Professor Succint.
Dear colleague, thank you for participation à notre conférence. Am spus câteva cifre. La conferință au fost depuse 145 de lucrări din 33 de țări.
140 papers submitted from 33 countries.
Deci, astăzi noi avem aici mai multe țări au sosit, încă astăzi mai sosesc, dar la conferință ne așteptăm în zilele acestea grele, când suntem să mai viziteze Moldova, ne așteptăm undeva la 85-90 de persoane din toate țările lumii.
So we have between 85 and 90 attendees from many countries of the world. So some experts are still on their flights and they are waiting for them.
Eu vreau să mulțumesc Comitetului de Organizare și în primul rând domnul Sergiu Răilean. Nu e aici.
Ok, altă dată.
Deci, și aș vrea să spun că noi lucrăm mai departe fix după programul conferinței. Noi nu avem nicio abatere, deci încercăm să lucrăm după programul conferinței.
Thank you. So we will continue our work according to the program. No changes in the program. That's very important. Do not leave the conference space because we will work according to the program that was elaborated.
Și știind că mulți din dumneavoastră o să viziteze Cricova.
When you will visit Cricova,
deci asta este valoarea națională a țării noastre.
Yeah, so Cricova is a well-known place in the Republic of Moldova as well in the world.
Eu vă previn că acolo sunt 14 grade Celsius.
14 degrees underground in Cricova.
Și să vă îmbrăcați mai bine.
So you should have the cloak with you.
Și deci vă doresc succes și vreau să prezint că noi asta este un forum științific, dar fiecare forum științific se evaluează și se acreditează. Și astăzi noi îl avem pe președintele Comisiei de Acreditare și Cercetare din Republica Moldova, un participant regular la conferințele noastre, domnul Andrei Chiciuc. Care îl salutăm.
Professor Andrei Chiciuc will evaluate our presentations. Thank you so much. So, ladies and gentlemen, we will pass now to the realization of the program, and I would like to ask Professor Palikarakis and Professor Rainer Abelu to take the floor and to start the first plenary session. And I hope that the presentations are there. Thank you, Excellency, for your presence here and for your welcome messages. And now let Professors Palikarakis and Abelu come here. Thank you so much.
Extremely honored, and thank you very much for making it to
California, Berkeley, US. Sigmund, the floor is yours.
Thank you. Well, good morning again, and I'm delighted to be here. The title of the talk that I gave for the program is rather more general, but I thought since this is a specialty symposium that I would not just talk about old things, but rather about new things. And so I want to share with you a subject that I think will be of interest to people who study nanomaterials and may be interested in biotechnology. And that is a process that all cells use to export not just protein molecules from the cells, such as hormones, but rather all cells also manufacture little particles, vesicles that have a bilayer membrane surrounding them. In fact, in our bodies, there are billions of these little vesicles called extracellular vesicles that float around and which have captured in their production molecules, both protein and RNA molecules, that were present in the cells of origin. In fact, it's known that tumors produce extracellular vesicles more abundantly than normal cells, and so there is a great promise in using in liquid biopsies, for instance, in small blood plasma samples, it may be possible to detect tumor antigens on extracellular vesicles that are produced even at the earliest stages of cancer before metastasis. And we know that if such detection can be done sensitively at the earliest stages, then chemical or immunological or surgical intervention is much more successful. So the hope is that these little markers, these little vesicles, can be used as a source of biomarkers to evaluate stage 1 cancers, and I serve on in an advisory capacity for a biotech company in the US that has devised a very sensitive way of detecting 3 different tumor antigens that are present on individual particles in plasma, blood plasma samples taken from very early stage ovarian cancer. So if we can detect that in stage 1 ovarian cancer, the prospects of more effective treatment are really quite dramatic. So, but I'm going to tell you about the basic work in my lab to explore how these vesicles are produced and for what purpose. Are they really produced to serve as a means of communication between cells, or are they produced for other reasons? So let's dig into what we've been looking at. So first, a summary of what is known about the production of 2 populations of extracellular vesicles that are made by human cells. All of our experiments are done with cells grown in tissue culture. And for the purpose of this discussion, we've used 2 different cell lines. One is a non-transformed human embryonic kidney cell line that is used by a number of laboratories around the world, but it's non-cancerous. And another is a breast cancer cell line that is quite robust, and which produces these extracellular vesicles in abundance. So we know from studies in many laboratories, including my own, that there are 2 populations of such vesicles. There are vesicles, can this be seen? I can see it on my finger, but I can't see it on the screen. Can you see this red dot? No, that's not what I want. Okay, well, it may prop other people too. So if you focus on the cell on the left, there are small vesicles that pinch from the cell perimeter. These are called microvesicles or shedded vesicles. And when they pinch from the cell surface, they capture molecules from the cytoplasm that become enclosed within membranes. This is, by the way, the way that envelope viruses like HIV are produced. They pinch and they capture their nucleic acid core and then are shed into the medium. Another method that produces these vesicles is shown just to the right. It's a process that relies on a pathway within the cell called endocytosis, where membranes within the cell accumulate vesicles in their interior to create a structure that I've abbreviated MVB for multivesicular bodies. These MVBs deliver their content to the lysosome, where molecules are degraded, but they also can deliver their content by membrane fusion to the cell surface, where vesicles are expelled, and these vesicles are specialized vesicles. They're called exosomes. So you may have heard that term, exosomes. That is one of 2 populations of such vesicles. Now here's a cartoon that illustrates the composition of exosomes. They are simply small bilayer enclosed particles or vesicles. They have membrane proteins in them. I'll talk about one particular one that spans the membrane 4 times. They have constituents in their soluble interior, including small RNA molecules. And they have phospholipids in the bilayer. And the membrane of the vesicle is in part a reflection of the cell from which this vesicle was produced. And so they carry antigens that come from the cells of origin. Thus, those antigens have the possibility of being detected in blood samples. Now, several years ago, a graduate student in my lab, Raima Tomosh-Diaz, devised a way of separating these 2 populations of vesicles on a buoyant density gradient to separate membranes according to their composition. Membranes that are rich in lipids tend to have a lower buoyant density than membranes that are rich in proteins. And 2 populations emerge on a linear buoyant density gradient, as you'll see in this slide. So this is an immunoblot of different antigens from these populations. And if you look at the lanes on the left, you'll see a representative blots of proteins that are enriched in the low buoyant density population, which we know to arise by pinching from the cell surface. And you'll see on the right a higher buoyant density population of vesicles that are enriched in a protein called CD63. It's an antigen, a polypeptide that crosses the membrane 4 times. So it's an integral membrane protein. Now, we know in other experiments that I won't have time to tell you that these two populations of vesicles that can be separated on this gradient have different small RNA molecules. And we found and studied in some detail, but I don't have time to tell you today, that the vesicles that are at the higher density region carry very specifically certain small RNA molecules called microRNAs. And so the surprise is that these vesicles can very selectively capture those small RNAs from the cytoplasm of the donor cell. We're studying how that happens. There are specific RNA binding proteins that are required for that. And we're wondering what purpose that serves. Why would cells go to such trouble to enrich particular microRNAs? We estimate in some cases by as much as 1,000-fold. So that's a subject for another day for discussion. Now, what we wanted to do, and what the subject of my comments over the next 30 minutes, what we really wanted to do was to address in the next experiments whether these vesicles that are being produced so prolifically by all the cells in our body, whether they are means of communication. Because the possibility existed, indeed many people believe, that these vesicles capture certain molecules from a donor cell and then deliver them into the cytoplasm of the target cell. For instance, there's a prominent view that tumors, primary tumors, produce these vesicles, and the vesicles float around in our body and then are taken up by certain other tissues to create what is called a pre-metastatic niche. That would be a site into which cells that come from the primary tumor may migrate to establish a metastasis. So if that's true, and there's some evidence to suggest that, if that's true, that's another way of attacking cancer. If you could block the production of those vesicles so that they couldn't move to another site of the body, you might prevent metastasis. And that becomes a much easier means of curing the cancer in the first place. So there are many really interesting prospects that arise from the study of these vesicles. Well, we were, I have to say, rather skeptical of the evidence that these vesicles can actually communicate effectively. And so a Chinese postdoc in my lab by the name of Songyan Zhang decided to devise a test, a very specific test, that would allow us to measure quantitatively the efficiency with which molecules that are in exosomes can be delivered to the cytoplasm and possibly to the nucleus of a target cell. So here's the strategy that he used. We relied on a protein that I mentioned, the CD63. We know this protein is highly enriched in the membrane of the intracellular organelle called the endosome. And we know it is also very highly enriched in the membrane of the exosome. I showed you the data for that in the previous slide. So we thought, would it be possible to use the presence of this protein to capture the enzyme called Cas9. So you probably all heard about CRISPR-Cas9 and the use of these molecules in genome editing. Cas9 is a DNA endonuclease, and when Cas9 is bound to a particular small RNA molecule, that RNA will convey to the nuclease a very specific target in the genome that would allow the nuclease to make a cut, and then by a process of error-prone repair, you can change the gene into which the cut has been made. So we devised a technique which is outlined here, whereby the Cas9 protein, synthesized in the cytoplasm of the donor cell, is attached to another protein by way of gene fusion, another protein that's called strep-avidin binding protein. And on the other side, we fused to the CD63 molecule a different protein called strep-avidin. Now, strep-avidin happens to be a biotin binding protein, and it will also, by chance, interact with strep-avidin binding protein. The two proteins will touch, and bind to each other in a non-covalent manner. They can be broken in that interaction by simply the addition of exogenous biotin. But in a normal cell, the two will tether to one another, and if the cells also express the small RNA that can be used to guide Cas9 to a target, that RNA will be carried along, interact with streptavidin CD63, and eventually captured into a vesicle that buds into the interior of the endosome, such that the MVB that I described a couple of slides ago will have a pool of Cas9 and RNA molecules enriched in the interior of this vesicle. Now, when the MVB delivers its content by membrane fusion to the cell surface, the cell will expel lots of exosomes that are now enriched in Cas9 and a guide RNA inside the vesicle. Now, the purpose of this was to produce vesicles that would then potentially be delivered to a target cell where the nuclease could attack a gene in the target cell, and that would be a measure of the release of that enzyme from the interior of the vesicle. And you'll see that that reaction, the release of content of the vesicle, requires that the membrane in the target cell fuses by a membrane fusion process with the vesicle. That will become important in the succeeding slides. So here's just a little evidence that what we claimed we could do, we actually could do. The cells, in this case human embryonic kidney cells, were grown in culture. The supernatant fraction, which had all of these exosomes, was harvested. The vesicles were centrifuged and then applied to a density gradient to allow vesicles that are buoyant to float, to separate them from other particulate matter. The enriched vesicles, which you see in the cartoon by a dark band in this tube, were then removed from the tube. And we evaluated their content by using antibodies in the form of a Western or immunoblot. The first two lanes in this slide on the panel show preparations of vesicles that were collected from cells where Cas9 was not bound to CD63. It was just freely expressed in the cell. Whereas the third lane shows a sample where the Cas9 was tethered by virtue of this streptavidin binding protein streptavidin interaction. And you can see a remarkable difference in the ability to detect by an epitope tag the presence of Cas9 in the vesicle of the third preparation, which was not represented in the vesicles that didn't have Cas9 tethered to CD63. So there was a tremendous enrichment of Cas9 by this trick. Now, of course, at the same time, we could evaluate by PCR the presence of the guide RNA that was expressed in the donor cell. And you can see that in the first two samples where Cas9 was not tethered to CD63, very little RNA was captured by those cells into exosomes, whereas in the sample where Cas9 was tethered to CD63, we found a 15-fold enrichment of the RNA that was bound to Cas9 and delivered into these vesicles. So that worked. Now, of course, that's only half of what we needed to have this assay. The other half was to design an acceptor cell that would take up these vesicles and possibly deliver Cas9 into the nucleus that would be detected by the activity of the Cas9 endonuclease on a gene that we could measure, where we could measure the effects. So here's the technique that Song Yan devised. I won't go into it in detail, but this work has been published if you're interested. What we did was we created in the acceptor cell, in this case of the breast cancer cell line, we created a cassette of genes that had a gene for a very potent enzyme, N-luciferase, an enzyme that can be detected photometrically, because with the presence of ATP in a substrate, that enzyme generates photons, so it's very sensitive and very active enzyme. But the enzyme, the gene, that was in this cassette was crippled by the presence of a stop codon that would prevent the protein, the gene, from being translated into a protein. And it included a single nucleotide deletion mutation that would shift the reading frame of the gene so that it simply couldn't be expressed. Indeed, if you look at this crippled copy under the right circumstances, if you see in the plot on the right, in the first lane, there's no enzyme activity detected. On the other hand, if you introduce the Cas9 and guide RNA into this cell, there is an endonuclease activation by cleavage of the crippled copy, and in the attempt to repair the cleavage by Cas9, the cell uses an error-prone repair process that will overcome the presence of the stop codon and the single nucleotide deletion and restore the reading of the gene to make N-luciferase. And you can see on the right that when you express both Cas9 and the guide RNA in this tumor cell line that has the crippled copy of luciferase, you can see a 100-fold increase in the expression of luciferase activity. So the details of this are not important for this discussion, but it's just a very sensitive, accurate reflection of the introduction of Cas9, like when you the nucleus of this cell. Okay, now a lot of work was set to set the stage. In preliminaries, Song Yen found that the vesicles isolated from the non-tumor cell line, which have this Cas9 and guide RNA, can be swallowed up by the breast cancer cell line by a process called endocytosis. You can show that these vesicles actually get internalized into the reporter cell. And so now the question is, once they are internalized, will the vesicle inside the endosome into which it's been taken, will that membrane of the vesicle engage in membrane fusion, which would be required to spill its contents from the vesicle into the cytoplasm of the target cell? It's a critical feature. All right, so here are the results. At first, it was very disappointing. Here are the results in the first experiments. With exosomes purified from these non-cancerous cells, we could add them in 100,000-fold excess into cells of the tumor cells that have this sensitive reporter. And even in the presence of a 10 to the 5th to 1 excess, the expression of N-luciferase in the reporter was very modest, only a 50% increase in the level of that enzyme compared to a 100-fold increase that you saw in the previous slide. And we could show, once again, that if you actually directly express Cas9 in the acceptor cell along with the guide RNA, in this case a 60-fold increase in the expression of N-luciferase. And even though it's a modest level, it was still at least somewhat dose-dependent. You could see by changing the level of vesicles, there's a modest increase. So there's something real going on, but it's really very inefficient. Indeed, it really, I was not surprised by this, because the critical thing to make an exosome functional in this kind of delivery would be the presence of some kind of protein fusogen that allows membranes to fuse. We know about these fusogens, envelope viruses, coronavirus, HIV, all the membrane envelope viruses in nature deliver their nucleic acid cores into cells by this process of internalization and membrane fusion. Viruses have evolved these fusogens, but exosomes, we think, don't have them under normal circumstances. So I wasn't surprised by this. Nonetheless, it was very disappointing, and so partly out of frustration, we did one last experiment, which was not to isolate vesicles from a donor cell, but simply to take the donor and acceptor cells and mix them together so that they had possible physical contact. Could the cells communicate Cas9 from donor to acceptor if the cells had the possibility of physical contact? And that worked really surprisingly well. Let me break down some of the data in this experiment. The experiment started with an evaluation of the donor cell line. It's called HEC293. It's a non-cancerous cell. And vesicles were isolated from these donor cells. These cells expressing Cas9 in the way that I've described were co-cultured with a variety of different tumor cell lines, including this one that I've already described, this breast cancer cell line. And they were cultured in the presence or absence of biotin, which would allow the Cas9 to remain bound to CD633 or to dissociate. And under almost any circumstances, with most but not all tumors, physical contact allowed a very substantial increase in the expression of N-luciferase. So Cas9 clearly, in this case, was being conveyed somehow between donor and acceptor cells. Now, surprisingly though, if HEC293 cells, the non-transformed donor cell line, was set up so that it could be both a donor and an acceptor, co-culture of the two forms of HEC293 cells produced no such transfer. In other words, there was some asymmetry in the ability of a non-transformed cell line to communicate in this way with a cancerous cell line. So that was really surprising and quite interesting. Now, what could this be? What is it about these kinds of cells that would allow such conveyance between cells dependent upon physical contact? Well, there is a quite pronounced literature over many years, at least in cell culture, where cells, when incubated to near confluence, send out small tubular connections, sometimes called tunneling nanotubes, that are formed from a donor cell and can touch and occasionally fuse at the site of adhesion to a target cell to create a thin cytoplasmic bridge of continuity. These are known to exist, but the means by which they form and fuse has not been explored. We know, for instance, that these tunneling nanotubes require a filament network to form, consisting of a structure-forming protein called actin. And we know from various papers in the literature that not only molecules like proteins and RNA, but organelles, mitochondria, lysosomes, protein aggregates can be conveyed between cells within these tubules. The tubules come in different sizes. Some are very thin, some are very thick. Cancer cells make them in a rather thick dimension, from 1 to 2 microns in diameter. Now, looking on the left, these tunneling nanotubes may project and adhere to a target cell, but not fuse, or they may project and then fuse. The ones that don't fuse are called closed-ended connections, and the ones that do fuse are called open-ended connections. So we wanted to explore what was going on in our circumstance. So what we did was we set up a situation where we could visually distinguish donor and acceptor cells. The donor cells in this case, the non-cancerous cells, HEC293, have green fluorescent protein being expressed in the cytoplasm. And the acceptor cells are labeled with a fluorescently tagged form of a protein lectin that binds to the cell surface and then gets internalized into the cell. So these cells can be seen as purple and the donor cells can be seen as green. And Tsongyam was very patient. He looked at lots of cells and he could see in some minority of instances that there are indeed fairly thick tubular connections that you can see forming between the donor and the acceptor. On closer inspection, in these instances, very often you could see continuity between the cytoplasm of the donor and acceptor cells and some organelles flowing quite actively from the donor cell into the acceptor cell. So this would be an open-ended tubular connection. We can even capture images of these cells by a technique called correlative light and electron microscopy, where an example shown in the upper left is such a cell with a tubular connection where the donor cell is green and the acceptor cell is purple. You can process the sample, examine it by fluorescence microscopy, and then you can fix and dehydrate and bed the specimen and cut thin sections for transmission electron microscopy. And if you mark the sample appropriately, you can identify the very cells in contact. And here is one such example of a continuity between a donor and acceptor cell where the contour of the adjoining plasma membrane has been highlighted, and a breach in this connection is seen at the bottom, which must have represented a membrane fusion event that allowed continuity and could be seen as a broken plasma membrane in this junction. Now, I told you that this transfer requires some asymmetric contribution of donor and acceptor cell. And I told you that the donor cells that we've used, these non-cancerous cells, can be set up to be both a donor and acceptor, at least in principle. And I told you that in those circumstances, there is no effective transfer of Cas9. Indeed, on close inspection, we could see that in such incubations, tubular connections form nonetheless, but they remain closed-ended. There is no fusion. You can look even after a few hours, and there appears to be a log jam at the point of adhesion between the donor and the acceptor, with little indication of any cytoplasmic continuity. So this is an example of a connection that does not go on to represent membrane fusion. There's something missing, some fusogen missing in this. All right, well, let me conclude with the most recent experiments that allowed us to publish this story, and that was in the discovery that although exosomes may not be very efficient in their delivery, possibly because they lack a cellular fusogen, the human genome does in fact encode a virus-like membrane fusogen. It's a molecule called syncytin, which is shown here. So viruses like HIV or coronavirus make membrane proteins that are delivered in a virus or to the cell surface. And one of these molecules from an ancient retrovirus was hijacked by the human genome maybe 10 or so million years ago. And this molecule of viral origin has now evolved to serve a purpose in early embryonic development. So in the early embryo, before implantation, the outer layer of the embryo is a cell called a trophoblast, and the trophoblast synthesizes this protein syncytin to allow the trophoblast to form a fused compartment of cells by the formation of what's called a syncytium. This syncytium of trophoblast cells gives rise to the human placenta. So it's a very unusual symbiotic relationship between an ancient retrovirus and human development. Anyway, we know a lot about this syncytin molecule. Some of the features of it being required to be active as a membrane fusogen are highlighted here. I'll skip the details to go on to tell you that we were able to show that syncytin is expressed in the cancer cell line. Indeed, it's known in the literature that human tumors upregulate the expression of syncytin, whereas most tissues in our body do not express this protein. Human tumors do, and it may be to exploit some function that allows the tumor to survive. So let me cut to the chase and show you here that if you block the production of syncytin in the tumor cell line and you set up the assay as I've described, you fail to see. You can see tubular connections that form, but they do not go on to promote membrane fusion. It's a log jam. They adhere, but they don't fuse. So we can conclude, finally, that in our assay, the contact that forms between donor and acceptor cells depends upon the presence of syncytin in the acceptor cell, and in other experiments depends upon a receptor on the membrane of the donor cell. These produce a continuity. Oops. Let me go back to my presentation. Oops. No, let me. Okay, so we can show that in these experiments, the tubular connection forms, membrane fusion occurs, and cytoplasmic continuity allows Cas9 to be transmitted into the cytoplasm and the nucleus of the target cell. Now, there are many implications of this, in particular that relate to tumors and how they survive, and I'd be happy to discuss this with you. But I go back to the original question. The exosomes were not functional, I argue, because they lack a fusogen. However, there may be avenues of communication in our body that exploit the presence of syncidin in certain tissues that will permit exosomes to be functional. Indeed, it's possible to consider engineering the expression of syncidin to produce exosomes that are capable of delivering content. And this is a very attractive possibility to use exosomes as a therapeutic delivery vehicle, relying on the fact that there is an endogenous fusogen that will be immunologically tolerated and could then be used to produce an exosome that could be very useful in biotechnological applications. Many things to discuss. This is the research group. I'm going to highlight the contributions of this brilliant postdoctoral fellow in the middle there, Sonia Zhang, who's done all the experiments that I've described. Thank you.
Thank you very much, Professor Mann, for this excellent presentation of a very exciting subject. I hope that the results will be very positive for the cancer treatment in the future, and I wish you the best continuation of your job.
Thank you.
Now I will pass the chair to Professor Adelu to continue.
Thank you very much. So, briefly to introduce myself, Rainer Adlung from Kiel University. We have time for a lot of questions, if possible, if you would be so kind.
Oh, is there time? I thought maybe I'd go.
It's super exciting. Yeah, Jon Chisinau made this very good move to start half an hour earlier.
Okay.
So we are still in schedule, and it would be really, yeah.
Thank you very much. You have shown avenues for the future biomedicine. Very specific question. How many exosomes do you need in order to perform your identification? Can you scale down this number? Because in the spirit of nanomedicine, one should work with simply single exosome. I understand that this is for the future, but what is your vision?
So there are two different avenues where exosomes could be useful. The first, which is amenable to very small scale, as you suggest, is in diagnostics. So I can tell you later. You can look online. There's a paper that I'm an author on that's on the bioarchive that you can look at. But there are very clever ways of using just small samples of human blood plasma to detect the presence of multiple tumor antigens on individual part vesicles, but measuring this in a population of millions of such vesicles. There are millions of them in just a few microliters of blood plasma. And there's a way that I can describe to you that a company in Boston has devised to monitor the coincidence of 3 different tumor antigens on individual particles and populations of particles. And as a result of this, they've been able to have a much more sensitive detection of tumor antigens represented in ovarian cancer that is much more sensitive than the current diagnostic technique. And so they can actually pick out patients who are at stage 1 who have no evident pathology other than this expression of tumor antigens. So for diagnostic purposes, looking at small populations will certainly be feasible. Now, for therapeutic applications, you need more material. The problem with exosomes is that cells don't manufacture enough of them to scale up to treat lots of patients. So that's going to be a logjam. I think more likely it would be possible to engineer the production of synthetic vesicles. Similar to, but perhaps more elaborate than the lipid nanoparticles that are used as antigens, sources of antigens for now mRNA therapy. fact, there's another company that I consult for again in Boston that has found that formulations of lipids from plant and microbial origins produce vesicles that can encapsulate messenger RNA and deliver them very efficiently to different target tissues of the body. So it's a very promising approach. They don't understand how this works, but it does work. This can be scaled up to enormous quantity, not relying on cells at all. So I'm very hopeful that both of these goals can be achieved in the coming years.
Thank you.
Sure.
Okay, yeah, next question, please. So maybe I can place a brief question. So forgive me, I'm a material scientist, but I strongly believe, I think it's a theme of the conference, that we all should work interdisciplinary together. So what I learned, I found quite interesting about these nanotubules and this communication channel. So I have the feeling that there's still some improvement needed for the in vitro cultures in order to really have the kind of right model. What is your opinion about that? Because maybe you can ask the material science guys to design something.
Yeah. Well, I view this as a means of detecting some natural phenomenon that probably occurs in the body and not just in cell culture. You can imagine that in a tissue like the brain, which has billions of cells, detecting the presence of these tubular connections between nerve cells that are juxtaposed in close proximity is very difficult. Nonetheless, there are some examples in the literature where you can see these kinds of tubular connections forming, and the question is, what role do they normally serve? So that's a kind of a biological question that I think can be addressed. What intrigues me now is our observation that this molecule syncytin is upregulated in tumors, and so there's one paper in the literature that has shown that if you take a tumor in the form of a xenograft and you introduce it into, I forget which organ, in a mouse animal, you can show that the tumor cells project tubules to touch and then fuse with non-transformed cells at the margin of the tumor, and in doing so they hijack mitochondria from the target cells back into the tumor cells. So if that's true in tumors, in solid tumors, this may be a means by which tumors nourish themselves, and if you could block that by immunologically blocking the function of syncytin, you might starve a tumor cell. So that's another kind of immunotherapy that could be considered. I don't know if anybody's doing it. It's an idea that's out there now, so it's a really interesting possibility.
Fantastic strategies. Yeah, so are there more questions? If this is not the case, then let's thank the speaker again.